So today it's what, Wednesday the 21st, right, of May, and we'll be picking up on the rest of Chapter 3, maybe a little bit of Chapter 4 as we go into today's lecture. Today is the last time I'll see you before our exam next Wednesday. Monday, of course, we're on holiday, and next Wednesday we have a full-length exam. That's all we'll do that day. What you'll need to bring with you will be a Scantron 884-E form. That's the two-sided green full-size form. Make sure you have a pencil with you and an eraser, and that's all you'll need that day. When you're done with the exam, you're done for the period. The exam is 100 questions. I suspect most of you will be done within the first hour. You will be allowed an hour and a half. Okay, I don't think you'll need it all, but you can certainly plan on being out of here about 5:30 or so at the latest on that day. So that'll be next Wednesday, and that exam will cover what? Chapter 1, which was the hierarchy and the atom-to-atom story and the different organ systems. Chapter and homeostasis and what anatomy and physiology are, those things. Chapter 2, over the chemistry. And that's the area that I just know from historical understanding that that's the area that students are more weak in. If you've had chemistry, you're probably fine. If you haven't had chemistry, that's the area you're most concerned about. But atoms and protons and neutrons, isotopes, understanding how electrons are involved with ionic and covalent bonds, that there's polar and non-polar covalent bonds, and then the conversation about carbohydrates, proteins, lipids, and nucleic acids. So that's Chapter 2. That's about a third of the exam. A third from Chapter 1, a third from Chapter 2, and a third from Lab 1. So that entire PowerPoint conversation during Lab 1 with body cavities and directional terms and planes and serous membranes, that conversation. Everything in Lab 1 except for parts of the microscope. That microscopy section is not on the exam, but everything else as far as the PowerPoint and the exercises that you did and the things you were quizzed on in that first lab quiz are also very important material. Now, that Lab 1 material is also found in Chapter 1 of your Martini book. Now, I want to apologize right now. I realize that in the pre-labs, it keeps referring to Saladin textbook. That's the old textbook we were using. So I apologize. I'll be updating that as we go through. But right now we're okay. There will be a little bit of a chapter number change as we go forward. It's not going to affect you. I'll just keep you tell you where we're going to be in the textbook. But there might be a couple of chapter changes along the way that look different in your note pack from what is reality in your textbook. But I'll keep you straight. Also on that exam, vocabulary, terms 1 through 20, A through cusp. And so I'll be putting those terms together. The exam is largely Scantron, multiple choice, but there will be some fill-in-the-blank questions, and those fill-in-the-blank questions will be the vocabulary, as well as a few images that you'll label some things. Now, what images do you think would be important on Exam 1, based on what we've done? Say it again. Planes, absolutely. Recognizing the body planes. Anatomical regions. Perhaps recognizing different body systems. If I showed you a picture of the endocrine system versus the lymphatic system versus the muscular system, would you be able to identify it? Okay, your directional terms. So there won't be many questions like that, but there will be a handful that you will do fill in the blank. But the vast majority of the exam will be multiple choice. Okay. I think that's about it, right? Everything I'm talking about today is not on the exam. Today's content is going to be part of your exam number 2. So let's go ahead and go back to the vocabulary, and then we'll pop back into a continuation of Chapter 3. I need to go to slide 16 with you today. And continue this through 20. So chromo. You guys are all way too young to remember Kodachrome. or Fujichrome, film. It was the film when cameras actually used film, right? And so chrome was color film. Or chromocytes would be cells that have a particular amount of pigment or color in them. Chyme, C-H-Y-M, refers to juice. When we get to the digestive system, we'll see that once nutrients reach your stomach and they're churned by the stomach acids and by the muscular action of your stomach, that what leaves your stomach no longer looks like what went in. It leaves as a pasty, liquidy stuff, juice called chyme. Side, spermicide, homicide, suicide. We've seen that side term to mean kill. Circum, around. Circumcise, circumduct, circumnavigate. Sero, yellow. If someone has cirrhosis of the liver, it's a condition, but what happens is that the liver, as it's malfunctioning, their eyes become yellow, and there's some yellowing of some of the tissues, and so that's where it gets its name. But sero meaning yellow. Size, to cut. Excise, to take it out. Clast, to break. There are cells called osteoclasts that we'll discuss in chapter 8 or 9 that have to do with breaking down bone. Co, with or together. And you'll see another term that means the same thing here in a moment, but to cooperate, to operate together, right? To operate with somebody. Seal, I know it looks like coal, but seal is a swelling or a hollow cavity. We are said to have a coelom, a hollow cavity in which all of our organs are found within our body. Com and con, there are the other two. So co, com, and con all mean the same thing, with or together. Contra, against or opposite. Contralateral, something on the opposite side, right? We saw contralateral in lab as one of our directional terms, contralateral versus ipsilateral. And corn, if you have a corn, you have an area of hardened skin. Corp, body. Corporal punishment, if you think, or corpus luteum. When we get to the reproductive system at the end, we'll discuss the corpus luteum, this yellow body that's found within the ovary. Cost, rib. Intercostal muscles, intercostal nerves are nerves or muscles between the ribs. Coxa, hip. We'll see the coccyx joint or the coxal joint referring to the hip joint later in the course. And crani, as in the cranium, referring to the skull. And then finally for this exam, 1 through 20, crine or crino, to mean secrete. You know that the endocrine system is characterized by a bunch of glands, all of which secrete hormones within the body, right? Endocrine, endo meaning within. We'll get to that later in the course. But endocrine. Creto, to separate. When we get to the blood, we'll see that one can do a hematocrit. And when you do a hematocrit, you are separating out the portions of the blood, one from another. Cunei, wedge. They're cuneiform bones in your body. They're wedge-shaped. And finally, cusp, meaning a point. Your bicuspid teeth have 2 points. Your tricuspid have 3. Okay. So that brings us to the end of the vocab for exam number 1. Questions on vocab? Are you concerned about it? A little bit. I think after the first exam, you'll say, okay, I've got this. Again, don't wait till next Tuesday to start studying this. I'm hoping that you have already made your note cards, 1 through 20. You're done with that. You were done with that last week. And that you have been flipping through that set of cards 5 minutes a day, once a day. And in that 1 minute, that 5 minutes a day, you've already got this vocabulary pretty much under control. So that when you see the word cusp again, you know that it means point. And when you see the word ambi again, you know that it means both sides. Okay? So it just becomes very natural for you, those vocabulary terms. And if you do wait till the night before, it won't be as successful. So I encourage you, this is a holiday weekend. That's a good and a bad thing. It's a good thing that we have Monday. Most of us have Monday off, no classes. You've got more time to relax. You've got more time to study, the choices you make, right? Are you going to study or just have a good time? Whatever you do, though, just give yourself a few minutes away from the family to open your books, look at your things, and spend a little bit of time each day this weekend, and I think you'll be very successful next Wednesday. The campus is closed on Monday, of course. Tuesday, Janae will be upstairs in the resource room, 249, as her hours are posted. She's also there on Wednesday. So if you had any last-second questions, you could always stop in and see her, see me on Tuesday or Wednesday during my office hours, drop me an email. Any of those ways are great to clarify anything. The way that I would approach this is as follows. You have your homework assignment. Well, first of all, you're coming to lecture. Thank you. Or review the lectures on YouTube. Is that working for everybody? Okay, I'm up to about, I don't know, 3,000 minutes. So people are watching this stuff. I don't know who you are, but you're watching the videos. And we get to watch all these statistics. It's kind of fun. And I know if you're on a tablet, I know if you're on a cell phone, I know you're on a computer. I've got all kinds of statistics to follow you watching those videos. So I would come to lecture and or listen to the videos, those who are hybrid. And then you've got your homework assignments to interact with, and I'll fix that so you can go back and redo them. And then you have those worksheets that I gave you. They may not have their worksheets. Everybody got that in lab or on Monday. So look at that. Again, if there's something in the worksheet that we have not directly discussed, don't worry about it, okay? There's more information in the book than what I've discussed. Those worksheets are not made by me, but by the publisher. So there may be a few things in there that we have not discussed. Just skip it, don't worry about it. And then there is a quiz on mastering as well. Now that quiz is due when? Monday at midnight. So it's due, a couple of days, a day and a half before the exam. And you only get one interaction with that quiz. It is a grade, and it will help you get ready for the exam. So do that quiz. Once the quiz due date comes and goes, then you'll be able to go back in and review your answers and see what you did to clean up anything for the exam.
Is the quiz timed? Like
the quiz is, I don't believe it's timed. I'm saying that it's been 10 days since I put it together. I don't think I put a time on it because I don't typically do that on my quizzes. If you get there and there's time, then it's timed. But I don't think it is. Okay, anybody taking it yet? Timed? No, not timed. There's the authority right there. Thank you. Okay, and what else do I want to tell you? Anything else that needs to be said? Yes, ma'am.
Open it and don't do anything with it.
You can come and go.
Oh, you can. Oh.
You can come and go. It's not timed. So you can come and go. It's open book. Right? There's not a reason not to do well on it. You might miss a couple things. That's okay. That's why you get to review it before the exam. But it is a grade, so you want to do well on that. You want to make at least 80% on your mastering assignments. Right? 80% is the minimum that you should walk out of those assignments feeling like you've mastered the material. I'd like to see you get more than that, but don't leave those homeworks without getting at least 80%, and I'll fix it so you can go back and do it again. Anything else? Okay, so let's get back into chapter 3, and I'll start this conversation out by reviewing the cell and reviewing the organelles, and we'll do that review with this image. So I'm just going to point, and you're going to report what organelle we're looking at, and at this point we're just naming them, and then as I go through this presentation today, you'll also learn what each of these organelles is doing in the cell. So as I point to these, tell me what I've got. That's the nucleus. The dark center is the nucleolus or the nucleolus. Outside of the nucleus, there's a series of membranes. They have little dots on them. They appear rough. That's the rough endoplasmic reticulum. Those little dots are actually ribosomes. Then there are other clusters of these membranes, usually further away, which do not have ribosomes associated with them. That is the smooth endoplasmic reticulum. There will be another series of membranes that look like folded ribbon candy, the Golgi apparatus, or just the Golgi. Keep in mind that it is named after a person, so it should always be capitalized. You'll see these bean-shaped organelles, mitochondria, mitochondrion, singular, mitochondria, plural. There could be 10, hundreds, thousands of mitochondria within a cell. Here there are some little green Lego block-looking things. Those are the centrioles. Throughout the cell, I can see strings. Those are proteins. Those are part of the cytoskeleton, okay, giving the cell structure. Also within the cell, I'm going to see a series of different sorts of vesicles. I won't be able to distinguish them one from another, but some of those would be, for example, lysosomes, some might be peroxisomes, other might be secretory vesicles. On the outside of the cell, there is a plasma membrane or a cell membrane. On the outside of the cell, I might see some long processes. These are cilia. I might see very, very short processes, microvilli, and if it were a sperm, I would see one single long flagellum, singular, right? We'll hear flagella, but flagella is plural, so a sperm has a single flagellum. So that's just identifying the organelles. That's good, we're on our way. And then the rest of our conversation today, we're learning more about these organelles and what they're doing for the cell. Any questions on just identifying? Remember, don't memorize color. The blue thing may not always be the nucleus, and the green thing may not always be the Golgi. But instead, look for their shape and their relative location one to another. So I'm going to spend most of my time talking about the plasma membrane as far as any of the organelles go. Any functional unit of the cell can be considered an organelle, and the membrane is that as well. Now in lab this week, I know we did a lot of things in lab. We talked about osmosis and diffusion, pH, solubility, tonicity, cell cycle, mitosis. There were a lot of concepts that were discussed and introduced in lab. Some of them were more familiar to you than others, and we'll be using those different concepts as we move through the semester. And in that very beginning of that presentation, although we were playing with auger plates and dyes, what I really want you starting to think about is that every cell in your body, all the trillions of cells in your body need their supply of oxygen and energy molecules, and every cell has to get rid of its waste products. And every one of those molecules needs to go in and out of the cell, in and out or through the plasma membrane. So the plasma membrane becomes the barrier, right, that keeps things in or out, or the gateway that does allow certain things in and out of the cell. And we'll see that this is highly controlled for the most part. Remember, we said that the membrane is selectively permeable. It only allows certain molecules in or out. However, there are some molecules that just are able to get in and out of the cell because the cell membrane is also considered to be semi-permeable. So it's both selectively and semi-permeable. So the membrane, what is it made up of? The cell membrane or the plasma membrane, same thing, is made up of a layer of, actually a double layer of phospholipids. I mentioned phospholipids as a form of lipids that was important. And when I introduced them, I kind of showed you the same idea. I showed you a round ball with two fatty acid chains. So it's the same idea, except now I've got thousands, millions of them lining up side by side. And if we think about it, what was the chemical makeup, or what were the characteristics of these fatty acids? Those fatty acid chains, all carbons and hydrogens, were said to be non-polar, right? Non-polar. And another way of saying non-polar is that they're not going to interact with what? Water. Remember, water's polar. And so these non-polar fatty acid chains that are part of the phospholipids are not going to easily interact with water, but they will interact with themselves. And so what happens is that these phospholipids line themselves up such that their lipid portions are facing toward the inside of the membrane. They're facing each other. Now, the outside of the membrane, these round gray spheres, would be those phosphate groups. And remember, the phosphate group has a charge on it. It has a negative charge on it. So those negative charges tell us that the phosphate group is polar, and that those phosphate groups can interact with water that would be both on the outside and water that's on the inside of the cell. So this phospholipid bilayer, this double layer, creates basically a bubble around the cell. But it's not just these phospholipids. There are also going to be a lot of, here it's called membrane channels, which is a particular type of protein. So these are proteins that are in the membrane. And it's through those membranes, those membrane proteins become basically a bridge, a port, a channel through which molecules can move. Now, the movement of molecules into and out of the cell, though, is not random. It is highly regulated. So there wouldn't just be these openings in the membrane that allow anything in or out, but there'd be thousands of different types of these proteins, each of which is specific for allowing a single type or a single group of molecules into or out of the cell. Now, the other word that I want to use for this is, for this inside part, all of this in here would be hydrophobic. So all of this stuff inside, where all those fatty acid chains are found, would be hydrophobic. Now, what does that word mean to us again? Hydrophobic means that those molecules are fearful of or do not associate with water. Whereas the outside, these phosphate heads, right, these phosphate heads out here are actually hydrophilic, right, which means that they have an affinity for or a love of or easily associate with water. And that's true not only on the inside, but also out here on the outside of the cell. So that's a basic, very basic cartoon of the phospholipid bilayer that makes up the cell membrane. If I went to Walmart and I bought some of these phospholipids, and I could see them, and I just dropped them into a beaker of water, they would have the opportunity perhaps also of setting up themselves like this, right? And all I'm showing you is that these molecules are going to associate themselves away from water. So all of those hydrophobic tails are going to create a sphere on the inside. And on the outside, all of those orange would be the phosphate heads, which themselves are negatively charged and would face out toward the water. So that's just simply the characteristic of all lipids, right? All lipids are non-polar. All lipids would not dissolve or are insoluble in water. So the plasma membrane I'm describing to you as having both phospholipids and proteins. So the membrane is not just one molecule, it's many, many molecules. The phospholipids are arranged as a double layer, 2 layers of these molecules. And remember, the phospholipids are unique in that they're partially polar and partially nonpolar, partially hydrophobic, partially hydrophilic. There's also going to be a fair amount of cholesterol in the membrane. Now, again, cholesterol gets a bad rap in our modern society as something we want to avoid, but we must have a relatively high concentration of cholesterol in our cell membranes. Without it, our cells cannot survive. So we have to have cholesterol. In fact, our liver makes cholesterol for us so that we can have plenty of it in our cell membranes. And what else did I tell you about cholesterol? I told you it was a lipid. It was under the steroids, remember? And I told you that the steroids were made from cholesterol. So not only do you need cholesterol for your cell membranes, but you also need cholesterol to make the sex hormones, the testosterone, the progesterone, the estrogen that you've heard of. Also in the cell membrane are glycolipids. Now, glycolipids, lipids is the core, so they are lipids, but they also have on their outside or associated with them a sugar group of some sort. So glycolipids, phospholipids, these are all molecules that are lipids that have some extra unit on them, like a phosphate group, or in this case, sugar groups. So that's the lipid component of membranes. But in addition, there's also going to be proteins. And I showed you those proteins as those green channels. And these proteins are going to come in two basic structures, integral proteins and peripheral proteins. As the name suggests, an integral protein is one that is integrated all the way through the membrane and would be visible from both the inside and the outside. It creates a true channel. Or some proteins are peripheral, that is, they're attached either to the inner or to the outer layer. So they would be hanging on one side or the other, but they would not be visible from both sides of the membrane. So let's take a look at a better picture in a moment, and also let's describe what these proteins are doing in the plasma membrane. Again, we've already talked about the idea that transport, molecules must go into and out of the cell. So we've got to have this way for molecules to move in or out, but again, it's selective. Each of these proteins allows only a certain type of molecule in or out. Then some of these proteins are for intercellular attachment. Inter means, we'll get to this later on, but inter means between. So that is, what are the attachments between the cells that help the cells to anchor one to another? So cells are grabbing onto each other through proteins found in the outer layer of the membrane. There's also anchorage for the cytoskeleton. Cytoskeleton are the proteins inside the cell that give the cell its overall shape. So some of those cytoskeletal proteins are also anchoring to the membrane proteins to give the cell its overall shape. And many of these proteins in the membrane are enzymes themselves. So they are going to be catalyzing a chemical reaction that occurs only when the reactants are near the surface of the cell. Cell recognition. How does a liver cell know that it's a liver cell, and how does the body tell a liver cell from a kidney cell? Or, better question, how do pharmaceutical companies create medications which are targeted to a certain body system? How do we make drugs that only affect our liver? How do we make drugs that only affect our brain or our kidney or our stomach? Well, different cell types have different proteins on the outside. And so the medications are made such that they recognize the liver or the kidney or the brain. And they recognize it because the outside of the cells have these different proteins, which we now understand are unique to each of the cell types. So we use those proteins as a way of targeting medications. And then how does a cell, remember we talked about living cells or living things react to their environment. Well, how is it that a cell reacts to hormones, or how is it that a cell reacts to chemicals in its environment? Well, there has to be proteins in the surface which are going to receive those signals, and when those signals are received, that signal is going to cause something to happen inside the cell. And so we've got this idea of signal transduction. This message is sent from one cell to the next and into cells. So these proteins are critical, right, for the overall functioning of the cell. Here's a more complete cartoon of the membrane. It's still very simplistic, but it's more complete than the last picture I showed you. In this particular image, the outside of the cell, out here, the extracellular fluid. Now, the other name for that extracellular fluid is interstitial fluid. You'll see that, so I want to make sure you understand that word. Interstitial fluid is a fluid found between our cells, outside of cells. Extracellular, outside of the cells. And then the blue down here would be intracellular, fluid within the cell. We see the membrane, so there's our bilayer, and the purple spheres are what? The purple spheres are the phosphopart, right? And then the long, little, skinny fatty acids, right? And there's two layers of them. So that means that the entire inside in here would be described as being hydrophobic, fearful of water, whereas the outside and inside, those purple spheres, would be polar, and therefore water would be right up against the inside and the outside of the membrane. We see some proteins in here. This protein is going into and out, means going through both layers. So that would be an integral protein. As with this one, this blue protein would be an integral protein because it's going completely through both layers of the membrane. This one looks to be a channel protein. Molecules can go through that opening into or out of the cell. Some of the other proteins in here, like this yellow guy, that protein is a peripheral protein, right? It's only present on the outside, or this one is a peripheral protein only visible on the inside. So again, peripheral versus integral proteins. I mentioned that different cells have different sugar groups or different molecules on the surface. This is one of those glycolipids. I should clear this off. See how there are these green molecules? Those are sugar groups, and those sugar groups are attached to the phospholipid. So that'd be a glycolipid, and this protein here, right, this would be a glycoprotein. It's a protein that has sugars hanging off from it. So there are lots and lots of different proteins associated with the membrane. And then finally, what we see is that there are all these cytoskeletal proteins down here within the cell, and some of those cytoskeletal proteins are attached to these proteins. That's what's going to help give the cell overall shape. So lots going on. Lots going on. Last thing. These yellow guys in here are cholesterol. So I told you that about 20% of the cell membrane is cholesterol. It has to have a significant amount of cholesterol to keep the cell membrane and its integrity. We've talked about this. The movement of molecules through the membrane, therefore, is selective. Only certain molecules can go in and out. So how do molecules go in and out then? Here are some choices. Number one, the molecules can go right through the phospholipid layers. A molecule can actually snake its way through or squiggle its way through, right through the membrane. For a molecule to do that, that molecule must be lipid soluble. Right? For a molecule to move all the way through the membrane and to interact with those fatty acid chains, that molecule must be able to integrate with those lipids, and we would say that molecule is lipid soluble. Or the molecule can move through a membrane channel, right, through a pore or a channel that's in the membrane. It could be carried by a transporter. Some molecules literally pick up and carry molecules inside the cell. And then there are vesicles, and we'll talk about vesicles in a moment too, where the cell reaches out and grabs things from the environment or allows things to be released from the cell as a big group of molecules. So let's take a look at this particular picture. There's a lot of information on here that I want to unpack. We're looking at the membrane. We're seeing the phospholipids. Right? So there's your phosphate head and your fatty acids bilayer. Let's say this is the outside of the cell. This is the extracellular out here and intracellular on the inside. And we have this integral protein, this red integral protein. And I see that this protein is specific. It's allowing these little red sphere molecules, whatever they are, to travel into the cell. Now notice out here, there are 7 of these molecules. On the inside, there are only 5. So the fact that there's a difference in the concentration of these molecules would set up a concentration gradient such that those molecules would move which way? By passive, right? By passive diffusion, those molecules would move from the outside to the inside because there are more molecules on the outside than there are on the inside. These molecules are being directed into the cell through this channel, and it's very particular. Let me go away from it and come back to that one. Look at these molecules. These molecules are not requiring a protein carrier. These molecules instead are going right through the membrane. They're being absorbed straight into the cell. And it tells us that these molecules are lipid soluble. So what does that mean to you, lipid soluble? So the plasma membrane is probably the one organelle I'm going to spend the most time on because it's so important to be thinking about how molecules get into and out of the cell, and it also helps us to understand that polar-nonpolar conversation. In lab, this week we did that little bit of solubility, right? Molecules that were polar could dissolve in water. Molecules that were nonpolar would instead dissolve in acetone or a nonpolar solvent. So think about that. If this conversation is not making sense, maybe that will help you pull it together as well. Now, the other organelles that I want to present to you, we can divide organelles up in different ways. Now, each organelle has a particular job that it does, a particular function within the cell, and different kinds of cells will have different arrangements or different concentrations of organelles. In other words, a muscle cell might look very different. Its organelles might be very different than the organelles of an epithelial cell. They'll also have a Golgi. They'll also have a nucleus. They'll also have endoplasmic reticulum. But the number of organelles might vary drastically. Now let's take a look at a way of organizing the organelles. We can do this a couple of different ways, but I'm going to first describe to you the organelles which have a membrane around them. So we're going to call these the membranous organelles, and then I'll finish up with the non-membranous organelles. So they're simply called the membranous organelles. Either they are largely made of membrane or they are surrounded by a membrane. And this is going to start off with the endoplasmic reticulum, both smooth and rough. It is largely a layer of membranes. Also, the Golgi apparatus is largely a layer of those ribbon-layered membranes. There's also the lysosome and the peroxisome. Those are small vesicle-type organelles which are surrounded by a phospholipid bilayer. And then the mitochondria itself, the mitochondria is unique. It's actually surrounded by a double membrane, a truly double membrane around the mitochondria. So let me start off by just going through these, a couple slides on each, a couple of cartoon images of each. So the rough endoplasmic reticulum, it is called rough because it looks like it would be studded or rough to your touch, and we now know that that roughness is the ribosomes that are tightly connected to this. Ribosomes are the key organelle for producing proteins. So whenever you think of the rough endoplasmic reticulum, you're thinking about an organelle that is specialized for the production of proteins. Whereas the smooth endoplasmic reticulum does not have ribosomes, therefore does not make proteins. It instead is specialized in producing steroids and fatty acids. So steroids and fatty acids are made at the smooth ER. Now, as an aside that we'll come back to later, muscle stores extra calcium in a similar structure to the smooth endoplasmic reticulum. But for now, all I want you to focus on is that the SER is producing steroids and fatty acids. Do you agree those are not proteins? Right? Steroids and fatty acids are both types of lipids. So lipid production. As a cartoon picture, again, in the upper left, you see all the dotted structures. Those are the ribosomes. That'd be rough ER. And then further away from the nucleus will be a cluster of membranes without ribosomes, the smooth ER. A few more functions just to look here. Again, it's a place where we're producing proteins. The smooth ER making steroids, making fatty acids, making phospholipids. Right, so the phospholipids are being made also at the smooth ER. And the other thing is that the smooth ER is detoxifying drugs and alcohol. So what type of cell in your body would you expect to have a high percentage of smooth ER?
Liver.
Right, the liver detoxifies and breaks down alcohol. And so we would expect a liver cell to have a higher number or more of smooth ER, as an example of somehow how some cells are different from others. Here's another cartoon of this. Again, the nucleus, the nucleolus, the nucleus, the nucleolus, and all of this around here would be the rough ER. Keep in mind that this is a cutaway, and that the rough ER would be going around the entire spherical nucleus, right? Not just in the planes that we're seeing, but it would be around the entire spherical center. And then further away, removed from the nucleus further, would be what I always think of, looks like underwater sponges of some sort, and those would be the smooth ER. On the bottom, this is a electron micrograph, and I know it's hard for you to appreciate it, but these membranes that we're seeing, those double membranes, those are the actual endoplasmic reticulum, and all these little dots that are gathered all around it are the ribosome. Next, the Golgi, again capitalized, named after somebody. The Golgi is going to be packaging, receiving and packaging those molecules that were made by both the rough and the smooth endoplasmic reticulum. Okay. So that means that the Golgi will be receiving the proteins made by the rough and receiving the lipids made by the smooth ER, and I always think of it as the UPS station of the cell. This is where different proteins are being packaged and distributed specifically to areas of the cell. So to me, the Golgi is the UPS or the FedEx center of the cell. It's receiving, packaging in a new way, and then sending off to its future location. Again, if we look at a cartoon representation of the Golgi, we see a bunch of flattened membranes. And it's interesting, they even use shipping terms to describe the Golgi. So on the side closest to the nucleus, this is the receiving side, the receiving region. And so proteins made on the ER are coming in to the Golgi, and they go through these layers. And as it goes through these layers, proteins are being packaged and redistributed, and then those proteins are going to emerge from the shipping side of the Golgi. That'll be closer to the membrane. And now those vesicles will send these molecules. Whatever this yellow molecule is, that molecule is now being shipped to a particular place within the cell. It could be staying in the cytoplasm. It could be going to the membrane to be released via exocytosis. It could be traveling back to the nucleus to go into the nucleus to affect DNA. But regardless, these proteins now are being sent to their particular place. So packaging and sorting and modifying. In this color-enhanced electron micrograph, this green would be what? That's the nuclear membrane. And if you look really closely at it, remember the nuclear membrane has gaps in it? Okay, so that's the nuclear membrane. That makes this the nucleus. And that makes this purple the what? Nucleolus, right? So there's your nucleolus, the nucleus, and then out here, the first membranes that I'm circling would be what? The rough ER, the endoplasmic reticulum. And then the flattened series of membranes even further away would be part of the Golgi. Okay? And molecules are being made, sent from the ER through the Golgi, and are being released on the shipping side of the Golgi. Another cartoon. I think this one may be a little easier to see because it's more late. Here is some of the rough ER over here. On the left-hand side, you see the brown. So there's your rough ER. And then the receiving side going through the layers of the Golgi and then out. And here you can see that Some of these proteins have been packaged to be exocytosed out of the cell. Here you see that some of these proteins have been packaged into a particular type of organelle called a lysosome. That would also be true of some peroxisomes. What's not shown here is that some of these vesicles would actually travel back and perhaps go all the way back to the nucleus.
Excuse me? I have a
Yes.
question. You said that they're packaging up like the proteins and the lipids, and then it can go straight to the plasma membrane. Does that mean that their membranes are all the same?
They are.
Can be used
Good point. So these membranes are interchangeably used. I showed you that one picture of exo or endocytosis where the membrane was being blebbed in and incorporated into the plasma membrane. That's exactly true. So good point. So these membranes, this is a phospholipid membrane. And look what's happening over here. So this membrane that's around this is now being incorporated and becoming part of the cell membrane. Turn this around, somewhere else on the cell, there would be what? There would be some things being endocytosed. And some of that membrane would now be breaking off and carrying things into the cell. So there's this, it's a really dynamic highway of molecules moving in and out. So good point, good clarification. As the Golgi is packaging these proteins and products, some of them are secretory vesicles. Now, secretory vesicles, as the name suggests, are going to be released from the cell, secreted out of the cell. But others of them will also form the lysosome and the peroxisome. So what lysosomes and peroxisomes are specialized collections of enzymes. Within a lysosome, there will be a certain set of enzymes. Within a peroxisome, there'll be a certain set of proteins and enzymes. So let's take a look at these two. We'll start off with lysosomes. What does the word lyse mean? To break, right, or to cut up. And zome, we'll see over and over this semester, means a body. So a lysosome is a body, a structure that is going to be breaking things down. Lysosomes are critical within your cell. They are the garbage men. They're the waste management system of your cell. And here's basically what happens. Here's a molecule. Whatever these molecules are, they're coming into the cell. And they're being brought in by what process? Endocytosis. Or maybe pinocytosis, but they're coming in, right? Phago or pinocytosis. They're coming into the cell. So we never talked about what happened, right? All I said is, oh, look, now what was on the outside is on the inside. So now these molecules that were on the outside of the cell are inside this little vesicle, this little vacuole. Well, what happens further along is that, just like soap bubbles, here's a lysosome, and this lysosome is going to bump into, either randomly or actively, we're not even sure if this is a random process or somehow is facilitated within the cell. But at some point, these molecules, this vesicle, will bump into a lysosome, and just like bubbles will, they will blub together. Now, the enzymes that are found inside the lysosome are going to break down whatever this molecule is into its individual pieces. So what you have now is a garbage system that breaks down, degrades to their most simple form, whatever it came in contact with them. The enzymes inside a lysosome can break down proteins, nucleic acids, lipids, and carbs. They are, across the board, able to break down every kind of molecule within your body. So there'd be proteases and lipases and molecules that can break down sugars as well.
Are they using that then? I mean, what's the purpose of the breakdown on, so, I mean, obviously it's quick in this picture, but.
Let's say this is an immune cell, and it just gobbled up a bacterium. Now, the purpose of that lysosome will be to destroy that bacterium or that parasite. If this was a molecule coming in from the neighborhood that could be used for nutrients, then the lysosome would be breaking it down into its individual pieces. Now, what I'm not showing here is that those molecules could then be used by the cell as building blocks for something else, or perhaps those molecules would simply be exocytosed and spit out of the cell, just as waste. Now, another process that the lysosome is important in is this term down here. So Tay-Sachs then is a disease of the lysosome being defective, and is common in Eastern European groups as well as, for example, Irish American groups. And what happens here is that there's an enzyme missing from the lysosome. That enzyme no longer is there, so fatty acid type molecules build up. And those fatty acid molecules begin to clutter up or gunk up the nervous system, slows down the ability of the nervous system to work properly. And as a result, death occurs. It's a pretty hideous death by age 4 or 5. So we care, right? We care about lysosomes because things can go wrong. So we want to learn about them. Okay, maybe in the future you'll have a patient who has some sort of Tay-Sachs disease. Or, and let me show you this picture of it. This is a peroxisome going on to the next one. Now, I can't tell a peroxisome from a lysosome, nor will I expect you to. But if you see a little vesicle and I point to it, it's either going to be a lysosome or a peroxisome, but you won't have to figure out the difference between the two. And as you look at the bottom of this slide, you see that, again, as I've already mentioned, peroxisomes detoxify. So there'll be a high number of them in liver cells. And what they do is they convert hydrogen peroxide into water. So they get this name peroxisomes because they're able to use peroxide in their breaking down of molecules. Now, peroxisomes also can go wrong. Anybody ever seen this movie? Okay, Lorenzos Oil. It's on Netflix. It's a good little movie. It's old now, 1993. It's about a couple who have a child with the disease. The disease was adrenoleukodystrophy, ALD. It's similar. It's a disease where there's a dietary problem, and they're trying to figure out a cure for their kid. And he's been diagnosed, and he's going to die of this disease unless they can figure this out. And the peroxisome also is breaking things down. And eventually with this disease, again, their nervous system starts to get shut down. The kids go blind. They lose their reflexes. This is typically a boy disease because of its transmission, its genetic transmission. But what this story is about is how they figured out how they could modify his diet. And if this child avoided certain oils, then this byproduct that was killing him would not accumulate, and he could have a relatively normal life. So it's a fun little chick flick, maybe a few teary moments, but along the way you'll learn something if you happen to watch it. But again, I pointed out because the whole story, I don't think they even say it. I don't think they even say the word peroxisome anywhere in the movie. But that's what's going on, right? It's a defect in the peroxisome. It's a genetic problem related to the peroxisomes. Okay. Last organelle that has a membrane around it is the mitochondrion. This is an important organelle because this is the one that makes the ATP. I mentioned in lecture 2, metabolism. Right? Metabolism is the collection of all of your cellular reactions, and if you look at the overall core of your metabolism, it's this reaction called cellular respiration, or these series of reactions referred to as cellular respiration. And what was cellular respiration? Remember what this was? Cellular respiration, right? Let me change colors. Cellular respiration. It was what? Sugar, right? Glucose, plus what? What do we do every day? We eat sugar. We get some oxygen, right? I'm not balancing this equation. But overall cellular respiration is what? Glucose plus water and oxygen. We also get some water in, right? We all drink water, we all breathe oxygen, we all eat sugar. As a result of that, what happens? Our body will make what? Carbon dioxide, which we blow off, and we'll make more water that we urinate and use in our metabolism. And out of this also we get a lot of energy. And that energy is in the form of ATP. Okay? Not all of this happens in the mitochondrion, but the last steps of this whole monstrous process does happen in the mitochondria. And it's in the mitochondria that we make the ATP. Okay, so with that understood, okay, let's go back. So the mitochondria are the powerhouse of the cell. They are the ones taking in oxygen, right? Taking in oxygen from the environment and using it to burn, if you will, burn that oxygen to burn the sugars with that oxygen to make the ATP. Every molecule of glucose or sugar that you bring into your body has the ability of making 36 or 38 or so ATP. I'm not going to go into that conversation. That's a conversation for a general bio course. You'll also get an entire lecture, exam over metabolism if you take microbiology. So the time will come for that story in greater detail. Just know that this is happening in the mitochondrion. If we look at a cartoon of a mitochondrion, we'll notice it doesn't have a single membrane, but it actually has a double membrane. There's an outer and an inner membrane, the details of which I'm not going to worry about right now, but it's within the membrane and within these spaces within the membrane that all the enzymes are there that form the ATP that we desperately need to run the machinery of our cells. Okay, so we need to have this ATP. And that's really all I'm going to say about mitochondria. Here's that overall story in a different way of looking at it. You're taking in some food, some candy, some sugar, whatever it is, and that's C6H12O6. There's your glucose molecule. And through the process, one of the processes is glycolysis. So break that word down. Glyco, sugar, lysis, right? The breakdown of sugar. And as we do this, as long as we have oxygen around and our mitochondria are working well, we will get lots and lots of ATP. That's the bottom line. Okay. If there is no oxygen around, well, if there's no oxygen around, we're dead, right? But at the cellular level, if there was no oxygen available to the cell, then instead of making lots of ATP, it will instead make lactic acid. And this is the story behind muscle burn, right? When you're exercising and your muscles in that moment don't have enough oxygen to keep up with the activity that they're doing, your body is saying to you, hey, dummy, we don't have enough oxygen here right now, and your muscles are building up lactic acid, and it's oftentimes, that burn, right, is a reason for you to stop exercising because you feel that burn. Now you can work through that burn, but we call that anaerobic respiration. You don't get much ATP out of it, only 2, so you don't get much energy out of it, but you do get that lactic acid buildup, and this all happens in the absence of O2. So when there's no oxygen around, we do anaerobic respiration. Anaerobic. Okay, and that's all I'm going to say about mitochondria. Again, all of the reactions in your body, all the chemical reactions in your body can collectively be called your metabolism. Right? Now, metabolism can be broken up into 2 parts. What are the 2 parts of your metabolism? If I were to say 2 parts of your metabolism is a combination of what? Your metabolism is equal to, you can't see that, can you? This will work better. Your metabolism is equal to all of your catabolic reactions, right? Or your catabolism plus your what? Anabolism. Your catabolism are all the reactions where things are being broken down, right? Whereas your anabolic reactions are all of the reactions in your body that are making things, right? Synthesizing things. So together, all of your catabolic reactions, all of your anabolic reactions, are going to make up the entirety of your metabolism or all of your metabolic reactions. Let's go through the last few organelles. These are the non-membranous organelles. They do not have a membrane associated with them. They're going to include things like the ribosomes. They don't have a membrane around them. It's going to include the cytoskeletal proteins. It's going to include the centrosome and the centrioles, the cilia, the flagella, and finally the microvilli. And we'll learn what each of these is doing for cells that have them. Start off with the ribosomes. Tiny, tiny, tiny little structures. Where did we already see them? We saw the ribosomes attached to the rough ER, but ribosomes are also just free floating around in the cytoplasm. Their major job is to make proteins. That is their job. Ribosomes are made up of two parts, a large and a small subunit. Nothing fancy there. And they look like little tinker toys. On the left-hand side is a cartoon representation of those two subunits, the smaller and the larger, nothing fancy. So the small and the large subunit, together they make the functional ribosome. This is that same electron micrograph I showed you before, all these little dots that are associated with the endoplasmic reticulum and some of these freely floating dots, those are all ribosomes. Let me go back to the statement at the bottom down here and make sure we understand this. What might be the difference between ribosomes that are on the endoplasmic reticulum versus freely floating ribosomes? This is a higher level question for sure. But ribosomes that are attached to the endoplasmic reticulum, as we saw a moment ago, what's happening? The endoplasmic reticulum is making proteins, and those proteins are going to be packaged in the Golgi, and those packaged proteins are oftentimes going to be sent out of the cell. If I were a protein and I was being made on a freely floating ribosome, then I would not be going to the Golgi. Right? So proteins that are made on freely floating ribosomes are not then going to go to the Golgi and be packaged. They're already just freely floating out in the cytoplasm. So a protein made on the ER is going to go to the Golgi and get packaged. A protein made on a free-floating ribosome in the cytoplasm is simply going to be a free-floating protein out in the cytoplasm as well. Cytoskeleton. There are three basic types of proteins that give the cell overall shape. Their names aren't that tough. Microtubules. Micro, small, tubes. If you were to hold a microtubule up, it's sort of like a toilet paper roll. Right? You can actually see down the center of it, there's a tube that's formed. The microtubules will be important in making flagella on the sperm and cilia. Microfilaments. Microfilaments, small filaments. Where, until a few years ago, would you find filaments in your home? Lights. Light bulbs, right? Those little tungsten filaments. So when you hear the word tubules, you're thinking of paper roll, paper product roll. When you're thinking about microfilaments, you're thinking about small, thin wires. Right? So very, very small, thin wire-like proteins. And they're going to be involved in muscle contraction and moving things and giving cells their structures. And then there's one that's intermediate in its shape or size, so they are called very unimaginatively the intermediate filaments. And we'll see how they provide mechanical support for the cell. I also want to go back up. Microtubules are also the proteins that make your mitotic spindles. We mentioned the mitotic spindles in mitosis. Those proteins that reach out and grab the chromosomes and pull them back during anaphase and telophase. So the microtubules are also the components that make up the mitotic spindle. Let's look at a cartoon of them. I would never ask you to recognize them, but I think it might help you just to get a visual on these. On the bottom, microtubules. You can see it's sort of a tube-like structure. You can actually see right down the middle of it. The thinnest of them are the microfilaments, and then intermediate in shape are the intermediate filaments. When you look at any cartoon, a nice cartoon of a cell, you'll see these proteins kind of all over the place inside the cell. Just know that they're giving shape and structure and support to the inside of the cell, giving it, overall structure. If you see something sticking out of a cell like a cilia or a microvilla or a flagella, you better believe that something is giving the cell that shape from the inside, and that's going to be some of these cytoskeletal proteins. Also, for motility, for the cell to move, flagellum and for cilia to move things around, they'd also be involved with that. And then this idea about cell division, right? These cytoskeletal proteins are important in mitosis. Looking at a picture of them, cilia. If a cell has cilia, there's never just one of them. They are very, very numerous on the surface of the cell. And the purpose of cilia are to move. Cilia are to me like wheat in Kansas in the summer. Right? It's this big field of these moving cilia, and they move in unison together. A cell that has a flagellum, and I really should put here, a single flagellum. When there's a flagellum, there's typically in our cells only one, and that's on the sperm, and that is for motility. And then microvilli are very, very small micro. Villus means hair, so a little tiny hair-like structure, and they're at the surface of the cell, and their job is to increase the surface area, to increase the size of the area of the membrane. You're never going to see these on the same cell. So cilia and microvilli are not on sperm. A cell that has cilia would not also have microvilli. They're going to be one or the other or none of them at all. So just to show a picture of this, on the bottom, there's the head of the sperm, and here's the long flagellum on the bottom. When it comes to cilia, cilia will be described as being found in the respiratory tract. So these are the cells lining your trachea and your windpipe, and this layer on the surface is mucus, and that mucus is helping to capture particles as we breathe in. Any kind of particulate or dust product is going to get caught up in that mucus, stuck inside our airways, and then with the beating of the cilia will beat in one direction, and the direction they're going to beat is moving it out of the body. So it's going to try desperately to get rid of that stuff and move it back up your trachea so that you can swallow it. Right? We're always swallowing those secretions coming up and out of our airways. What happens, though, with a smoker is that after a while, the body gets so sick and tired of those particles coming in, it gives up. And the cell stops making these cilia, and now those particulates in the cigarette smoke can make it all the way down into the lung, and 20 years later we call that cancer. Right? So the body basically says, screw this, I can't keep fighting this. So they cough and cough and cough, and after a while, the body just stops making these cilia altogether, and the body can no longer fight off those particles getting into the lungs. Last one, centrioles. It looks like some sort of little tinker toy. Now, each of these is a centriole. Each of these is a centriole, and two centrioles make a centrosome. I know you'll see those two words. They kind of get mixed up. But anybody know what OLE means on the end of a word? A centriole means something small. O-L-E, right? An arteriole is a small artery. So it's a small structure. So I always remember the centriole is the smaller of the two, and there are two centrioles that collectively make up the whole body, the centrosome, right? Centrosome. Now these centrioles were important because it's from these, I'm going to just kind of draw them like this, but it's from the centrioles that we create the mitotic spindles that are important for mitosis. And they are themselves made up of microtubules. So this is microtubules, that's what makes up centrioles, and microtubules are what emanate out from the centrioles to grab the chromosomes during mitosis. Very important. If a cell does not have centrioles, it cannot divide. If a cell does not have the ability to make mitotic spindles, there's no way the cell can divide and go through mitosis. That brings us back to the nucleus. Okay, back to the nucleus as an organelle. And we remember that the nucleus, let me back up. The nucleus does have a membrane. So I just stopped with the organelles, right? I went through the list. So the centriole was the last of the organelles that was a non-membranous organelle. Now we're just coming back to the nucleus at the very last bit of this. And the nucleus, of course, is a membrane organelle. It does have a membrane around it. Remember, it has those pores within the membrane. And within the nucleus, what do we find? What's inside the nucleus? The chromosomes, the DNA. And when we were looking at cells in lab undergoing mitosis, we saw those chromosomes. We saw them lining up during metaphase, and we saw them separating during anaphase. And it is the DNA and the chromosomes that contain all of the instructions for making your proteins and making everything else in your cell. So it's definitely the command center, and we know all about the nucleolus. Now I want to go back. What does the nucleolus do for the cell? I mentioned it in passing long ago, but what is the job of the nucleolus? Anybody remember? It said somewhere that the nucleolus was necessary for making ribosomal RNA, little r, capital RNA. Now, we don't know what ribosomal RNA is yet, but it sounds like it must have some important job in making what? Ribosomes. You're not wrong, right? So ribosomal RNA is a building block to making ribosomes. Ribosomes are necessary for making proteins. Does that help you understand why maybe the nucleus has to have pores? It has to have pretty big gaping openings for ribosomes to go in, or I should say to get out. Because what we're going to see is that the nucleolus is inside the nucleus, right? And it's going to make the building blocks for the ribosomes, but where are the ribosomes found? They're out here in the cytoplasm, right? So the major building blocks for ribosomes have to get out of the nucleus into the cytoplasm. They have to have a way to get there. So they're going to pass through these nuclear pores. And then where do they go? Many of them are attached right here to the rough ER. Not all of them, right? Some of the ribosomes are what? Freely floating. They're just sitting out here in the cytoplasm. They're not necessarily attached to the rough ER. Okay. Oh, I got ahead of myself. So here's what I said in words just a moment ago. Now look at this word, nucleoli. That is plural for what? Nucleoli is plural for nucleolus. Okay, not to be confused with nuclei, which is plural for nucleus. Okay, they're really, really close. So nucleoli is plural for nucleoluses, right? And nuclei, plural for nucleus. And again, I've already mentioned to you that the nucleolus is all about making ribosomes. What else is in the nucleus? Chromosomes. So chromosomes are these molecules. We saw them in passing when I discussed DNA and RNA. And you saw the DNA molecule was wrapped tightly around some proteins that then wrapped around and looked like a telephone cord